Domestication of Potatoes Due to Allele Diversity by April Hanaclaw, Alejandro Hernandez, Chelsea Sanders, and Michael McLeod. The potato, or Solanum tuberosum, is one of the most important agricultural crops in the world. Potatoes develop tubers from underground stems called stolons. This process is called tuberization. Originating in the Andean regions of South America, potatoes have a short day-dependent tuber formation as a vegetative propagation strategy and therefore will not make tubers in the long day conditions of spring and summer in northern latitudes. The potato plant maturity phenotype is a major genetic focus that is linked to chromosome 5. This phenotype varies through multiple genetic interactions and is responsible for several important subtraits, including tuberization conditions. For simplification purposes, we will describe these genetic interactions using alleles 1, 2, and 3, proteins F, A, and G, and C genes to model their effect on tuberization in potatoes. So what do these genes, proteins, and alleles do? Allele 1 is associated with late tuberization, while alleles 2 and 3 are associated with early tuberization. The F protein is a clock regulator and works in conjunction with allele 1 to downregulate C genes. The A protein is key in inducing tuberization, while the G protein, when overexpressed, suppresses the function of the A protein. Lastly, when the C genes are expressed, they act as a suppressor for the G protein. In the protein with alleles 2 and 3, a normal 5' to 3' gene sequence forms, which allows for normal protein formation and therefore regular functionality. In the proteins with allele 1, however, the 5' to 3' gene sequence contains a mutation in the polypeptide bond. As we learned in class, this can be due to missense, silent, frame shift, or nonsense mutations. Since this specific mutation leads to a truncated protein, it is most likely due to a nonsense mutation, which is defined by an early stop codon in the sequence.
So what genetic interactions delay potato tuberization to support growth in long day conditions? Allele 1 contains a binding terminal for the F protein, which destabilizes the allele. This prevents the promotion of C genes and therefore an excess of G proteins, which repress the A proteins. The y-axis represents the fold change, or presence of the protein in question, relative to the other alleles, which is directly related to tuberization. The x-axis displays the time of day, with the white area being the time frame in consideration, i.e. short or long. Finally, the title at the top indicates the protein being recorded. As seen in this graph, allele 1 tuberizes around late afternoon in the presence of G proteins under long day conditions, whereas alleles 2 and 3 fail to tuberize at all under long day conditions. Similarly, in the presence of the G protein, the wild-type plant, denoted WT, peaks its fold change later in the afternoon, while the plant with overexpressed allele 2 remains undetected. As demonstrated by the graphs, allele 1 initiates potato tuberization later in the day and is therefore supported by long day conditions. Then what promotes potato tuberization under short day conditions? Alleles 2 and 3 lack the binding terminal, therefore allowing C genes to accumulate. The C genes repress the G protein and therefore indirectly promote the A protein and ultimately early tuberization. As seen in the graph, in presence of protein A, allele 1 has no fold change, indicating no initiation of tuberization. Alleles 2 and 3, however, demonstrate fold change throughout the designated time period, aligning with our understanding of their protein A promotion under short day conditions. As seen in this graph, alleles 2 and 3 peak earlier in the day, while the fold change in allele 1 occurs later in the day. It is also important to note that under short day conditions, alleles 2 and 3 have more than double the amount of fold change than allele 1. As seen in the graphs, alleles 2 and 3 initiate tuberization earlier in the day and are therefore supported by short day conditions.
So what have we learned? Alleles 2 and 3 act as indirect inducers of protein A and therefore early tuberization, as seen by the dotted line arrow. Allele 1, however, contains a binding terminal for the F protein, which destabilizes the allele. This causes a lack of C genes and therefore an excess of G proteins, which suppress the A proteins, causing late tuberization. In summary, this study showed that these key interactions between the identified alleles, proteins, and genes were ultimately responsible for the time and success of tuberization in potatoes. It also demonstrates how our current artificial selection lab can be applied to real-world scenarios. By selecting the potatoes which express certain genes, farmers are able to maximize their harvests in climates all over the world.